macromolecules, and we're going to be looking at proteins. So proteins are made up of amino acid monomers. The pieces that make them up, the monomers, are individual amino acids. They're going to be held together. The bond that holds these amino acids together is called a peptide bond, and therefore, when you have a polymer of amino acids, you will have a polypeptide. They are composed of they team up together, and so if you have these polypeptides, which again is a chain of amino acid monomers held together with peptide bonds, one of these polypeptides could be what we call a protein, or like with many proteins, there could be several polypeptides together that actually form a protein. So the protein may be a single polypeptide or multiple polypeptides working together. These polypeptides, this chain of amino acids, they're going to fold together, and this is going to give them a 3D shape. So think of a chain of amino acids, be like any other chain. You have this linear conformation, but then it is going to fold in on itself, giving it a very particular shape that allows it to carry out its function, and that is going to make your protein, either a single chain or maybe you have multiple chains working together, and they will all fold up together and form this protein. So the monomers that come together to form the polypeptide, the amino acids, their structure is essentially a central carbon that is connected to two functional groups, as well as a hydrogen, and then this other part here. So you'll recognize the functional group on the left here as the amino functional group, the one on the right as the carboxyl functional group, plain old hydrogen also attached to this central carbon, and then importantly there is this R group, and so R is not a functional group nor is it an element. This R represents something that will be there depending on which amino acid you're dealing with. So change out this R group, one thing for another thing, and you end up with different amino acids. So we call this the side chain for the amino acid. So note that the carboxyl group has acidic properties, which means it can lose its hydrogen, and the amine group has basic properties, so it can actually gain its hydrogen. So it's as if this hydrogen comes off and then is attached over here to the amine group. That's essentially what's happening there. The name of the amino acid, once it has actually ionized, so it's that this hydrogen has come off of the hydroxyl group and is over here on the amine group, it's called a zwitterion, great name for it. And again, whether you're looking at the ionized version or not the ionized version, not a big deal for what we're doing, but you may see it as the unionized version here, where the carboxyl group is complete, or you may see it as the zwitterion version, where the hydrogen has left and gone to the other side. Not a big deal either way. So the bond that's going to be holding these together, so here's looking at the zwitterion version of it, but essentially we need to take an oxygen, a hydrogen, and another hydrogen away from these two amino acids. And so, and if you want to think of it as not being the ionized version, you can imagine the hydrogen is there instead of here. But either way, you're going to be taking an oxygen and a hydrogen, so depending on whether you want to look at it as the ionized version or not, you are getting water out of this. You're putting these two amino acids together, so it is a dehydration synthesis. That's close enough. And we are forming the bond between them, which is going to be called a peptide bond. So two amino acids are joined through dehydration synthesis, and we get a peptide bond out of it, and water. Here are 20 common amino acids. They're not the only ones that exist, but these are the main groups of them. And as you can see, again, this is drawing the ionized version. If you don't like that, you can feel free to move the hydrogen back over to the carboxyl group on the one side. But you'll notice that highlighted in here is the R group. So if the R group, if that thing that's attached to the carbon is just a plain old hydrogen, the amino acid you're dealing with is glycine. If it's a methyl group, then it's alanine. And so you can see, depending on what that R group is, each one of these is different, and that's what leads to the difference in the amino acids. And there are different types. There are ones that don't have polarity to them. The R groups are nonpolar. There's some R groups that are polar, and then there's some electrically charged ion versions of them as well, both acidic and basic. So different properties that these R groups can have. When the chain is completed, so you keep adding on amino acids, you keep getting them connected together with dehydration synthesis, forming the peptide bond, they are then going to be able to fold in on themselves. So imagine a chain that has certain groups that are going to be attracted to other groups and maybe not so attracted to different groups, and it's going to all fold up on itself, think like a magnetic necklace type sort of thing, where they're going to all fold up, and they will end up with a particular shape to them. And that shape, as well as the properties of the parts of that shape, are what determine the function of that protein. So this organization can be categorized into 4 different levels. First one is the primary structure, and really this is just the order of amino acids. We saw the different types of amino acids that you could have. Whatever order they happen to be assembled in, that is the primary structure. So maybe they would go with a glycine first and then an alanine next, or whatever the order is of those amino acids, that's the primary structure. The secondary structure is that those amino acids that are now connected together as a chain, they can start hydrogen bonding with the ones that are near them. And so there's sort of this initial folding that happens, and there's 2 ways that it happens. It forms a helix, or it sort of forms these more linear pleated sheets. We'll look at some pictures of those in a minute. The tertiary structure is next, and this is where the R groups come in. So remember we said some are polar, some are non-polar. So for example, if you have 2 polar R groups, they're going to be attracted to each other. They're going to want to come together. If you have 2 non-polar R groups or a bunch of non-polar R groups, they might be sort of forced in the middle away from the aqueous environment that the protein is in. And so the tertiary structure is where the R groups are playing. The sheets that are formed and the helixes that are formed in there are going to be held up in that arrangement with a tertiary structure as well. And again, big play with the hydrophobic polarity or hydrophilicness of it, depending on that polarity and the aqueous environment that it's in. There's going to be charges as well. We mentioned how we have some charged R groups in there, and they're going to be coming together. And there can also be some other things that will help them to fold up together based on the protein that you're dealing with. So these chaperones that help them to fold into their correct shape. Last of all, there's the quaternary structure, the 4th level, and this is where you've made, so you put the amino acids in order. They then will sort of fold in on themselves initially through hydrogen bonding. So here's imagine a chain sort of getting attracted to itself in a nice linear sheet type arrangement, and then the R groups can start interacting, and so it's going to get a little bit more globby, and you have this sort of 3-dimensional shape with the tertiary structure. Then if you have this one with a tertiary shape and this other, so that's one polypeptide, and then you could have this other polypeptide with its particular shape, they could team up together, and that would be the quaternary structure where multiple polypeptides come together. And then you might have your functional protein. You might have a functional protein after the tertiary structure where you just have one polypeptide, but often proteins are going to be made of several polypeptides. So visualizing that, again, imagine each one of these as your amino acid. So we have 1 amino acid, 2 amino acid, 3 amino acid, 4, and so on, and they're all just together as a chain. Then those chains can, depending on their polarity, they may form a helix type structure, or they might sort of line up with these sheet-like arrangements instead. So that's the secondary structure. And again, this is the polarity of the backbone itself, not yet the R groups. Then, oh, sorry, here's some more pictures actually. This is the helixes that are created between the backbone of it. And so it is the amine groups and the carboxyl groups within the backbone itself that are going to be attracted to each other and can form these alpha helixes. Or they can form pleated sheets. And so you can end up with these chains essentially being hydrogen bonded to another. So here's part of the chain that sort of folds in on itself, and there's hydrogen bonding across to another portion of the chain. So I'm trying to draw the lines as the hydrogen bonds, and you end up with these sheets forming. We see that with spider silk. The tertiary structure is then when the R groups start interacting, and again, they might be all polar, or a group of them might be polar, and so this particular chain right here and this particular chain right here, their R groups might be attracted to each other, so they're going to clump up together. Whereas like these ones here and these ones here might not be attracted, so they don't clump up together. And this is going to give us our tertiary structure. And then quaternary structure, again, where we have one polypeptide and another polypeptide getting attracted to each other through the polarities with them and the shapes within them, and then they form this overall functional protein. So think of this like what the Power Rangers do. They have their own individual units, but then they could team up together with multiple units coming together to forming a functional protein or Megazord. Again, quick overview of all of them together. Primary structure is the order of the amino acids. Secondary structure is the backbone hydrogen bonding to itself, which can happen in sort of far away ones or closer together ones, which would give you a helix or a pleated sheet. Then the R groups are going to start interacting, giving you this big globby polypeptide, and occasionally you may have more than one polypeptide teaming up together to give you a functional protein. That's the quaternary structure. Last, these structures are really important for their functions, and they are dependent on the conditions that they are in. So if you change the pH, the temperature, the concentration, the ions of the solution that it's in, these will disrupt all of that interaction that we had forming their structures, the hydrogen bonding, the polarities, and the hydrophilic and hydrophobic R groups. All of that's going to be different if the conditions in which the protein are different, and therefore the shape is going to change. So we refer to this as denaturation. They get denatured when you change their conditions. If you take a protein and you heat it up, its shape will change. If you put a different pH, its shape will change, and therefore it no longer has its particular functional shape. It may change to some other arrangement, including sort of coming undone, and this is what we call denaturation. And we do this intentionally with proteins when we do want to denature proteins. We can do that things when we're like curing meat or cooking. All of these changes in conditions are going to denature those proteins. Within a living system, it's really important that those conditions are maintained so that this does not happen.